Hello and welcome to this BioProcess International Ask the Expert webcast. I'm your host, Leah Rosen, the online editor for BioProcess International. Before we get started, just a couple of notes. This webcast is being recorded and will be made available for replay in the multimedia section of our website. We've muted the audio lines, but we welcome you to type in your questions for our speaker in the chat window on your screen. After the presentation, we will begin the question and answer portion, and I will ask our speaker your questions from the chat window. Your questions in the chat window will only be visible to myself and our speaker. So thank you for joining us today. It is now my pleasure to introduce our speaker, Anshuman Shukla from Intertek.
Hello, everyone. I'm Anshuman Shukla. I'm a principal scientist at Intertek Pharmaceutical Services, Manchester. And today I would be talking about the current analytical approaches to biophysical characterization in a regulatory environment. So I've structured the talk into 5 different sections. Section 1 deals with complexity of biopharmaceuticals. In section 2, when we move on, we're going to figure out what we are looking for, what we need to find out. The next section up would be the need for all of these biophysical characterization studies. Then section 4 would be based on biophysical techniques, the techniques that are available. And I'd end up with a brief data that I have generated on the NIST monoclonal antibody. But first, something about Intertek Pharmaceutical Services at Manchester, UK. We are an FTSE 100 listed company and part of Intertek PLC. We've got laboratories all across the globe, and we provide pharmaceutical testing across all dosage forms. Specialize in complex delivery systems and combination products and support both small and large molecules here at the Center of Excellence for Biologics Characterization in Manchester. So, I'll discuss the complexity associated with biopharmaceuticals, and by biopharmaceuticals I mean peptides and proteins. And I'll be talking about protein folding from the next slide onwards, but this slide is just to show that this is a very complex process and should ideally be taking a very long time. So most of us know that a protein is, once it's translated, it's a long polypeptide and then it folds. So we'll just look at the complexity of that. So if we just consider a very small basic 100 residue polypeptide, and we assume that each residue can adopt only 3 conformations. So, and I mean, there can be a lot more, but assuming there's only 3, there are 5 times 10 exponential 47 possible conformations possible. And then if we further assume that if it takes only 1 picosecond to convert from one conformation to another, a total time taken to find the right conformation would be 1.6 times 10 exponential 28 years. And to put that into perspective, this timeline is like 10 exponential 18 years more than the lifetime of the universe we live in. However, in spite of such an odd against its folding over these timelines, the process usually takes less than a second. So, moving on to protein folding. What is protein folding? So if we look at a protein after translation, and I'm not going into co-translational or post-translational folding, we just are considering a very basic scenario where a polypeptide, when it's synthesized, it's got, so the beads here represent the amino acids that are joined together by peptide bonds, and this folds into a globular form. And this actually doesn't happen in one step. So from an unfolded state to a folded state, it works through different folding intermediates. So the folding of a protein is a very dynamic process with formation of different types of weak non-covalent bonds such as ionic bonds, hydrogen bonds, and van der Waals interactions. And since these are non-covalent bonds, they are also in a constant state of rupturing and reforming, and a folded protein molecule is therefore in a very dynamic conformational state. And the figure at the bottom right corner here shows just the bonds and the complexity of a protein molecule That's how complex it can be. Another driving force in protein folding is the presence of hydrophobic amino acid residues. And since proteins are generally present in an aqueous environment, these hydrophobic residues are usually buried in the folded core of the protein and are also considered to be one of the driving forces towards fully folded protein. So, having discussed this, I'll be coming back to this later, but we will briefly discuss the manufacturing process of a biopharmaceutical product, where a protein is in a stressful environment during the different stages of production. And this dynamic protein structure becomes very susceptible to unnatural structural changes and can actually misfold. So the next slide on, I've got just a small video showing how this protein, this polypeptide chain, is looking at different possible combinations and folds into its native form. So it looks like a long time, but this was taken only over 1.5 nanoseconds. And so far, we have only spoken about a single protein molecule on its own. But if we consider that in a biological system, there are other molecules and chemicals present along with it. So in addition to trying to find the right conformation, a protein molecule is also in contact with all that is present in its surrounding environment. This has massive implications, particularly in pharmaceutical industry, where different formulations are tried to maximize protein activity and shelf life. If the conformation of a protein gets compromised, it might affect its stability and activity. So, and this is where biophysical analysis come into picture, because most of these techniques are non-invasive, in most of the cases anyway, and provide us with the relevant information that we need to know. What do we find out with biophysical analysis? So, we have spoken about protein folding and what does it lead to. So following translation, what we get is a primary structure, which is essentially the amino acid sequence. Now, this sequence undergoes recurring rearrangement to provide recurring structural motifs along a polypeptide chain. These motifs could be alpha helices or beta sheets, and they are referred to as the secondary structure of a protein. So once these are formed, these motifs start interacting with each other, and they form a 3D globular shape of a protein, which is called the tertiary structure of a protein. And if there are more than one polypeptide chains associated with each other to form a dimer, trimer, or whatever order of oligomer there is, it's the quaternary structure of a protein. And collectively, biophysical analysis provide information about the secondary, tertiary, and quaternary structure, which are also referred to as the higher order structures of a protein. So I've briefly mentioned protein unfolding. So if we move on to the next slide, we need to also define what is protein unfolding. So unfolding, a loss of 3D structure of a folded protein sufficient enough to cause any loss of function is called protein unfolding. It is not necessary that the protein has to be completely unfolded, as the image here shows. It's just an illustration. It does not have to be completely unfolded to be called unfolded protein. These unfolded proteins can still show localized, partially folded states. It is also important to note that even minor conformational changes in certain parts of the protein can destabilize the entire structure. Now, if you look at the figure on the right here, disordered protein. Now, this is put it in here to further emphasize on the complexity of protein folding and unfolding, because there are proteins that are intrinsically disordered and can only form a higher order structure when they come in contact with a ligand or a target molecule. Now, this is another challenge because all proteins do have some structured regions, even the fully folded 3D globular proteins. These regions are called random coil, and these could also be linked to the stability and activity of proteins. Therefore, the distinction between an unfolded and an intrinsically disordered protein is not very great. Another issue arising out of protein unfolding is protein aggregation. So, aggregation is anything which is an oligomer of the protein that is present in solution but is not expected to be present there. Now, these aggregates could be really small and soluble and are not usually visible to the naked eye, or they could actually be very large, insoluble, visible aggregates. It can be caused by the way different amino acids interact with each other, and which can cause a conformational change in any of the part of the protein folded structure. And this is a very relevant part of a biopharmaceutical characterization because it might affect biological function, its shelf life, its activity. It can elicit immunogenic response in patients. So, unfolding of a protein and aggregation could be caused by temperature. Now, because with temperature, there could be conformational changes that can promote aggregation. Temperature can accelerate chemical reactions such as deamidation or oxidation. It could be caused by freezing, thawing. So repeated freeze-thaw cycles can cause complex chemical and physical changes, such as it forms a creation of, ice solution interfaces. There could be a differential concentration of proteins in solutes. There could be pH changes due to crystallization of buffers. Unfolding and aggregation could be caused by stress caused due to agitation, which could be stirring, shaking, even if anything as small as just moving it from one location to another, transportation. It could be due to shear forces that it experiences during, say, chromatographic processes. It could be caused by the effect of solvents, the surface and solvents effects. Which could be like changes in pH, ionic strength, excipients. So if you consider something like changes in pH, it will affect the electrostatic interactions because it's affecting the charge in the protein. If you consider something like an excipient like urea, which affects the hydrophobic core of a protein and destabilizes it. It could be caused by changes in protein concentration. A lot of the proteins are known to aggregate at higher concentrations, which is most likely due to macromolecular crowding that can cause a change in conformation and protein unfolding, misfolding, or aggregation. So I previously mentioned briefly about the manufacture of a biopharmaceutical, that the protein is under a lot of stress during that entire process. And this is again a very simple representation of what might be happening. So in this scenario, if you look at it, I've cloned the protein, expressed it, I've let the cells grow, I've induced the protein, I've recovered the protein using filtration or centrifugation, purified it by chromatography-based methods, and then characterize it and do stability studies on it. So in any of these steps, if there is a change, it might actually affect the conformation of a protein. So such that I might clone it in a different vector with a different tag, or the way it's been expressed in a different cell system, which could be growing in a different growth media. There could be change in the way I'm doing chromatography, so it could be different binding and elution conditions. There could be different methods used, reagents, reference standards. So any of these might be able to actually destabilize the protein and cause it to unfold. So this brings me to the next part of my topic, where we're going to discuss why do we need it. So we have so far seen that there could be many reasons for a protein to lose its activity, which is good enough reason to go for as much characterization as possible, especially if you're developing a biosimilar. However, there are regulatory requirements as well that ask a submitter to support the application with relevant data. So ICH Q6P provides detailed test procedures and acceptance criteria for biological products, which covers both drug substances and drug products. And these guidelines are adopted by different regulatory bodies, and they refer to complete characterization, including physicochemical characterization that can include structural characterization and conformational studies of physicochemical properties. They also require purity and impurity analysis. Impurities, aggregates are one of the major impurities in biopharmaceutical products, and a test of biological activity or potency is also needed. But most importantly, they require orthogonal methods for most of these tests. So orthogonal methods are required because one test might lack the precision, sensitivity, or selectivity required for that analysis. If we consider section 2.2.1 of ICHQ 5e, it clearly says that it might be appropriate to apply more than one analytical procedure to evaluate the same quality attribute, such as molecular weight, impurities, secondary or tertiary structures. In such a case, each method should employ different physicochemical or biological principle to collect data for the same parameter. ICH Q6A also refers to use of new evolving technologies where they are considered to offer additional assurance of quality. ICH Q1A R2 ICH Q1A R2 requires assessment of stability and evolution of product shelf life. So that's another requirement for going for all of these tests. If you look at ICH Q6P, it requires assessment of spectroscopic profiles by UV, circular dichroism, NMR, or any other suitable technique. So there is therefore a need for characterization using as many analytical procedures as possible. So this brings me on to the next part of my talk where these are some of the biophysical techniques that are available and are routinely used. So not everyone uses every single technique, but a lot of the people use the same techniques. And of these techniques, we currently offer the techniques listed here. So for secondary structure analysis, we offer far UV CD or FTIR. For tertiary structure analysis, we offer near UV CD, fluorescence spectroscopy, or derivative UV spectroscopy. And we offer AUC, SECMOS, and DLS for aggregation analysis. For thermal study or thermal stability analysis, we offer differential scanning calorimetry. So I had spoken about requirement for orthogonality earlier, and this is just an illustration that CD and FTIR are both powerful methods, but using just one method for all sorts of studies is not going to provide us the answer, because we might miss some things if we use just one technique. For instance, CD is more sensitive to alpha helix content, so it will not detect as much beta sheet as would say FTIR does, which will, because it's sensitive to beta sheet, it will not detect more alpha helices. The limitation of CD is also if the sample is very concentrated, it's not very possible every single time because beyond a certain concentration, it's not possible. But FTIR, on the other hand, it struggles with low concentration of samples. Different formulations and different buffers, different excipients present in a sample might interfere with CD analysis, which again in FTIR is not an issue. And if there are particulates present in solution, there could be scattering of light, and that can cause problems. But this is not an issue in FTIR, where the main issue is actually strong water absorption in the amide 1 region that is used for secondary structure estimates. The other thing is about orthogonality is AUC, SECMOS, and TLS. All three techniques are brilliant in their own regard, and every technique has its own limitation. So with AUC, there's no limitation in terms of the mass range it can be studied in. It can be studied in its sample buffer or formulation of interest. There's no such thing like a chromatographic media where it could be running down a column and experiencing shear forces, which is the issue with segmolds, and it can or cannot cause conformational changes. There is a very good mass resolution in AUC. We can look at non-covalent or weak associations using sedimentation equilibrium and can get information about particle shape or conformation. However, the limitation is it's very low throughput and takes several hours to quite a few days. In segmolds, the sample gets diluted as it is injected onto the column, and the analysis might not necessarily be done in the same formulation as the sample is originally present in. And with DLS, the main advantage is that if we are looking at very small quantities of really large aggregates, it will detect it. But because of its low resolution, it's going to struggle if the mass resolution or the size distribution range is not big enough. So all in all, I mean, if we do just AUC, we'll not get the right answer. We might not get the right answer with segmolds or DLS on its own, or we might get it. So it's best always to actually go for as much as possible to do so that we can get the maximum information. So biophysical techniques in general are good for looking at higher order structural integrity and stability, protein aggregation and protein-protein interactions. They are really useful in pre-formulation where you can actually beforehand and analyze them in the environment they're going to experience, or they're useful in formulation where you have formulated them and you can study them in the proteins, in the buffers and additives of interest. So the last part of the talk is about some representative data on the NIST monoclonal antibody. This is not a comprehensive set of data. It's just acquired over a period of 3 days, and it's there just to show that even though these techniques are low throughput, the results can be obtained really quickly. So CD, the top panel here shows the far UV CD spectrum. So if somebody who knows and does CD regularly, they will look at it and they'll say, oh, that's a beta sheet protein. It's predominantly beta sheet. So we know that secondary structure composition of this is a beta sheet. And this is confirmed by when the deconvolution of this spectrum is done and we're getting 36% beta sheet in there. Near UV CD spectrum, which is the bottom panel here, is linked to the tertiary structure of the protein, and it can be used like a fingerprint. So any changes in the interior hydrophobic core that might affect the surface of the protein can be picked out by using near UV CD. This is the FTIR spectra of the protein, and again, the main peak here is being shown at 1636, which corresponds to an intramolecular beta sheet. And this is just a second derivative spectrum, which picks out more peaks that again confirm that this is a predominantly beta sheet protein and beta turns. SVAUC results show that there is one main peak at about more than 90% predominance with a certain S value, and this is the monomer. But we also see some dimer and trimer in low predominance. Now, if you look at the DLS data, there are 3 figures here. We have size distribution by intensity, volume, and number. Now, intensity is the one where we go in to pick out very small quantities of large aggregates. But if you look, so volume and number, they pick out the actual number of molecules present in the solution. But the results of all 3 here are the same, and they pick out just one peak with 100% predominance. So this is where I say that if the mass range is very close together, DLS has that limitation of not being able to pick it out. DSC of the NIST map is also very interesting because what we see here is the first transition at 70 degrees. That's corresponding to the CH2 domain of the antibody. That corresponds to the CH3 domain of the antibody. And that's the FAB domain. Generally for a protein, the FAB domain unfolds before the CH3 domain. So this again can be used as a fingerprint for the NIST maps and an indicator of its stability. The last thing I want to show is, and that is not used a lot in industry, is the derivative spectroscopy. So in this case, I've done UV spectrum between 300 and 190 of the RMRE, which is a NIST certified standard. And I've done a first derivative spectrum just to pick out the actual peak distribution. So this is a very powerful technique because it can be applied to simple spectra containing unresolved bands, and it picks out, especially in the second derivative spectrum, it picks out the unresolved bands very clearly. So it is very useful when there is interference from substances that are apart from the analyzed present, so say multi-component mixtures. And at Intertek Pharmaceutical Services, we do all of these analysis in a regulatory environment pertaining to your phase of development from preclinical phase characterization studies through to GMP and beyond. And these are the total services that we offer. Thank you.
Great, thanks, Anshuman. So the first question is, for an orthogonal approach to study secondary structure, for example, how do you decide which are the most suitable techniques?
So essentially, it depends on the molecule that's being studied, the formulation it is in, the concentration at which it is. So again, like I said, there's no hard and fast single method that would say is useful for it or more powerful than the other one. So if, say, the sample concentration is high enough, it would actually be better to use FTIR, or if there are excipients that can interfere with absorbance spectroscopy, so CD won't be suitable, it would be better to use FTIR. But if it is at a low concentration, FTIR might not be. So it's a case-by-case basis where we can actually pick and choose what technique is suitable for that particular set of samples.
Okay. And what is the typical timeline for a validation of DLS or CD method?
Usually, if it's not a complicated sample or not a complicated study, it's 4 to 6 weeks for a full validation. But again, that's a case-by-case basis because sometimes there's some optimizations that might be needed, so a pre-validation study needs to be done. So in which case, it can be longer than that.
Okay. And what would be the minimum level of characterization you would recommend if someone does not require all the techniques?
Again, ideally, if it's a biopharmaceutical, we should be looking at aggregation profiles. So analytical ultracentrifugation should definitely be done. And in terms of secondary structural studies, I would say, because this gives us information about both secondary and tertiary and quaternary structures. So secondary structure and tertiary quaternary structure studies should be done. So AUC and CD or FTIR, depending upon the sample.
Okay. For the audience, if you guys have any additional questions, you can see Anshuman's contact information there on the screen. The recorded version of this webcast will be available for on-demand viewing on our website, and as a registered attendee, you'll receive a follow-up email providing you with a direct link. We look forward to having you join us at future BioProcess International Ask the Expert webcasts. Look for those announcements in your inbox. Thanks. Goodbye.